Good morning once again, all the participants. Dear teacher friends, please excuse me. We are not giving any time break in between the two sessions because of some technical issues. So I hope you understand. Okay. So we have one more expert with us. She's none other than Professor M.S. Nalini. She is from Botany Department, University of Mysore. Presently, she is working as a professor in Botany Department. She has been graduated, post-graduated, and did her PhD from Mysore University only. Professor Nalini Madam, area of research is plant virology. She is expertise in plant virology. And the field of specializations are angiosperm morphology, morphogenesis, and tissue culture. She is also interested with bioprospecting of medicinal plants and microbial endophytes. Madam so far guided 3 PhDs and 5 MPhil students, and 15 students have completed project work at post-graduation level. Madam has published more than 32 papers. All the papers are presented and published in international journals. And she has also contributed 2 book chapters. And at various conferences in national and international level, she has presented her research findings in different conferences. So visited United States on academic assignments and visited Mysore Botanical Garden in St. Louis, United States of America. Besides all these, she is also member to various academic bodies and associated with the microbiologists, Indian Science Congress, and others. And of course, with her interest in research, she has brought funds from different organizations like DST, MHRD, IOE, and UGC. With this brief introduction, I am not taking much time. So I welcome once again, Madam, Dr. Nalini, Madam, and over to Madam. Thank you, participants. Okay.
Good morning, one and all. I want to hear your response. Good morning.
Good morning, ma'am. Very good.
Good morning, ma'am.
Good morning, ma'am.
Yes, respected coordinator of 15th refresher course in life sciences and dear participants. I'm here to present my topic, which will be displayed now. It's on natural products and their applications. The talk goes like this: applications of diverse natural products as sources of drugs against humanity ailments. So sir made it very, very specific that I should talk on the diseases and the applications in life sciences. So to introduce the topic, once and for all, we were also participants like you many years back, staff college, to deliver the talk, okay? And today, my thing is revolving around the contents of my presentation, which is of course on diversity of natural products and their importance as drugs. So schemes in natural product discovery, the update on the types of natural products as drugs, plants, which have given so many drugs for us, phenomenal drugs that have been discovered in the treatment of human-related diseases, microbes as the sources of drugs, marine sources of drugs, which has revolutionized the drug discovery program, approved drugs from marine sources, which are already approved and they are under clinical trials, and the conclusions that are drawn. Okay. Yeah. So to start, let us start with the theme of today's life science, that is innovations and applications of life science for human advancement. So life sciences comprise fields of science involving the study of living organisms, which includes both plants, animals, and humans. And biology refers to the centerpiece of all studies and recent innovations in molecular biology and technological advances in biotechnology have led to the development of new fields of specializations which are interrelated to us. Okay. So we have a definition of natural product, which are abbreviated as NPs. They have played a very crucial role in drug discovery program, whether it is cancer, ulcer, infectious microbial diseases or protozoan diseases, and also in therapeutic areas of cardiovascular diseases as well as the multiple sclerosis. Okay, so NPs offer both the conventional synthetic molecules, which are challenging for drug discovery process. So NPs have certain types of characterizations with structural complexity, higher molecular mass. They have to obey the Lipinski's rule of 5, which was proposed by the chemist Christopher Lipinski in 1997. And all those like hydrogen bond acceptors and donors, which would be equal to 5 or greater than 5, and partition coefficients are all characteristics that will make an oral drug competent. Okay, so that's why it's very important to learn about the rules before proceeding for a drug discovery program. And we all have all these information about how a natural product library or research works. Okay, we have typical extracts that are taken from either plant sources or microbial sources, and then you have to proceed further. So I have put forth a traditional bioactivity guided isolation step in natural product discovery. So the process starts with the obtaining crude extracts, that is from either plant sources or microbial organisms. So organisms, when you talk, it may be fungi, bacteria, actinobacteria, actinomycetes. All of them are producers of metabolites. So you need to ferment them, isolate them, ferment them. Then there are multiple rounds of fractionation steps, okay, in order to obtain the single compounds. Otherwise, we also have, when you obtain compounds or stages of fractionation, you need to evaluate the bioactivity. That is using certain assays. And if it obeys, you proceed further. So this is the basic step. Okay. Otherwise, you also have natural products with advanced analytical technologies that could be adapted in order to screen for a program. For example, I have given here the isolation of a bacterial strain that produces a unique antibiotic and its analogs. Okay, so you identify the gene cluster that is called as the biosynthetic gene cluster, and then proceed further rather than a redundant program such as screening without any target-based assays. Okay, so using this, many such actinomycins or the antibiotics produced by bacteria, marine bacteria, as well as the endophytic bacteria have been identified, which have been the source of drugs. Okay. Otherwise, there is a sequence-based metagenomics approach where you can use a particular character like the calcium binding domain or any other target-based assays and use a transformation system like we have Streptomyces silicolors, Streptomyces albus as organisms to which gene can be introduced, and then the choice of the metabolite can be made to be produced at a precise pace. And therefore, using this principle, an antibiotic malacidin has been identified with a unique functional group. So R refers to either you can put A and B are the analogs, either methane or methyl or ethyl, like that, okay, or methoxy, like that. So this is one approach. Otherwise, we have advanced genomic sequencing methods, okay, identifying the gene clusters from actinobacteria, which can give rise to definite antibiotics. Most of the Japanese antibiotics that have been developed by Japanese workers is based on the novelty of the strategy employed. They have I think 29% of the all antibiotics produced are from the extracts of actinomycetes, whether it is soil derived, whether it is endophytic, or whether it is from the marine environment. So we have several strategies like iChip method, which is a device to obtain soils from the sample and then cultivate the bacteria and then identify the metabolite, which are resulted in the identification of a novel bacteria, that is Eleftheria terrae, which has shown good antimicrobial activity against Staphylococcus aureus. And the compound obtained has certain folds of activity exhibited by this organism. Okay, so like that, you have many of the procedures in natural product discoveries, which could be the semi-synthetic analogs or synthetic analogs for modifying the bioactivity of a compound, okay, through derivatization process. So you can either change the bonding or remove the H group hydrogen from the oxy radical and then put a methyl group or methoxy to enhance the bioactivity of the organism. That is called fold discovery. Okay, and many of these examples are there in literature. I don't have to go to this. Okay, now when I was preparing for the talk, I got a very important information that was updated in 2020 regarding the sources of natural products as drugs from the past 4 decades. So this is a data that has been published by well-known natural product scientists, Newman and Craig. So if you refer their articles, every 5 years they have updated the knowledge on the botanical drugs or drugs from other sources. And then they have compiled a review as to know till 38 and 3/4 year, how many drugs are available from varied sources, okay? And that I'm going to present now. They have used certain codes in the analysis. B, for example, is a biological macromolecule. N is a natural product which is unaltered. Then NB refers to drug of botanical importance, natural product derivative. So like that, okay? Or NM, which is known as a mimic, and the vaccines that are used. Okay, so like this, certain subcategories of the terms have been made from 1997 till date, and it's still being used. Okay, now let us come to the actual approved drugs from 81 till September 90. That is before this publication was made. So a large chunk, that is N is equal to 1881, refers to the number of drugs that they have taken and then classified them accordingly as biological macromolecule, natural product derivative, etc. So you can see a large chunk is represented by the synthetic. Okay, so chemists have a great role to play here. The same data is published in a bar graph, which shows that probably in 1997 and 2018, there are a great percentage of drugs have come to the market. So similarly, we also have small molecule approved drugs, wherein again, synthetic molecules play an important role. And all this is necessary because we are actually contemplating the use of drugs in the treatment of human ailments. So according to us, an ailment is a manifestation of a disease. And there are several classifications in this. We know cardiovascular, we know respiratory, we know Alzheimer's and all of them. But if you take a list that has been supplied, so you know there are more than 50 different ailments, and many of these drugs function for all these ailments. They are in the market, okay? And if you take the indications of disease versus number of drugs, so for all diseases, there is a total of 464 drugs that have been approved. For 4 or more, that is 397, and the graph shows 3 or less, they are 70, equal to 70. So like that, the drugs are classified into several kinds, including the antimicrobial agents, wherein you can see that either the the green one represented by derivative of natural product or the synthetic drug plays a very important role, okay? And like that, antimicrobial agents can be classified into antibacterial drugs, antifungal drugs, and antiviral drugs. And by this graph, you can make out the large share of the chunk which is in the market. There is a list also, but since I have many more slides I couldn't put, so I thought the pie graph representation from the paper would be very apt for presentation. Similarly, we have antiparasitic drugs. Many of them are in the market. And
ma'am, excuse me, ma'am. I cannot zoom to see the things, ma'am.
Where I'm under presentation, you have been sent, right?
Oh, you will send it? Okay, ma'am. Okay.
Yeah.
Thank you, ma'am.
I'm not seeing it.
I can see it, but I couldn't zoom it, ma'am, to see the data.
Okay. I think the presentation will be sent later. So,
okay, ma'am. Thank you.
Yeah. So we have a classification of several types of drugs, anti-cancer drugs that have been in the market. Okay, from 1946 to 1980, that is quite a few years of gap. And then, you know, the data, each pie chart represents the number of N that is taken. That is N is equal to 247. So that means so many drugs are categorized as biological molecules, the natural product derivative, or natural botanicals, vaccines, like that. So it is self-explanatory that many of these drugs are approved, okay? And there is a list that have been given in the paper about the approved anti-cancer drugs. And 2017 and 18, you can find there are less than 25 drugs that have been approved in the consecutive years, okay? So like this. The data goes on, but due to time constraint, I don't think so I can present everything. Then we have anti-diabetic drugs. We have inhibitors of enzymes, alpha amylase and alpha glucosidase. And in this, biological macromolecules, such as the enzyme inhibitors play an important role, which is expressed in blue. And then similarly for multiple sclerosis and anti-glaucoma also, there are drugs in the market. So let's now understand the plant-based sources as drugs, okay? I guess you are all from different backgrounds.
Hello? Yes, ma'am.
Yeah, so botany, zoology, biochemistry, are they included here? Yeah. Okay. So now we know that plants have novelty and they are pharmacologically important because many blockbuster drugs have been directly or indirectly derived from plants. And as the data goes, at the dawn of 21st century, 11% of the 252 drugs that are basics and essential by WHO are defined as DoS with extremely flowering plant origin. And therefore, it is used in the treatment of human diseases, what I have expressed a few minutes back, okay? And though there are new approaches to drug discovery programs, such as combinatorial chemistry and computer-based molecular simulations and all of them, it depends on what best product you're giving to the individual or for marketable ability. And it seems that 50% of the approved drugs during the last 30 years are either directly or indirectly derived from natural products, especially for cancer, okay? And there is a great controversy in the data, but then as suggested by Newman and Craig in 2012, plants are still, or natural products still are employed with novel structures as sources of drugs, and the search continues, okay? And there are about 35,000 to 70,000 plant species in the world which have been screened for medicinal purposes, which is actually the drug discovery program. And 80% of 122 plant-derived drugs have been related to the ethnopharmacological purposes. That is, specific communities still employ plants as sources of medicine. And that is the main thing for drug discovery program. And the Food and Drug Administration of US and European Medical Agency, EMA, approves drugs of plant-based choices such as paclitaxel and morphine for treatment of the particular cancers and other related ailments to relieve the pain, etc. Okay, and yeah, let us just remove this slide. Because about a third of approved drugs in the past 20 years are based on the natural products or their derivatives. So this has been updated by Craig and Ed. Now let's go into the importance of plant-based drugs. The first one is the discovery of quinine for the treatment of malaria fever. Okay, so everybody knows the discovery is from the bark of a tree, Cinchona officinalis. Okay, and this tree is preserved from the Amazonian forest in the Manu National Park in Peru. So there are two compounds which are obtained from the Cinchona bark. One is quinine, the other one is quinidine. So both differ only in the slight removal of the replacement of the hydroxyl group and the hydrogen bond, which is here in the molecule in the quinol ring. Okay, so it was first isolated or extracted from the bark of the Southern American Cinchona tree by two French chemists. And it became actually a boon for the treatment of intermittent fever throughout the world. And Woodward and Doring in 1944 achieved the artificial synthesis of quinine. And it's one of the drugs that is used against Plasmodium species, as well as it has fever reducing ability. Okay, though several analogs such as chloroquine have been developed based on the structure of quinine, there is a resistance that is developed by species of Plasmodium, and therefore quinine is still used. Okay, and quinidine, a distereoisomer of quinine, is also extracted from the bark and is known to have a bitter taste, but it also can lower the irregular heartbeat or few heartbeat that is there. Another drug of importance is aspirin. So it is acetyl salicylic acid. So salicylic acid, when it combines with acetate, it's an esterification reaction which gives rise to acetyl salicylic acid. It's a non-steroidal anti-inflammatory drug used to relieve pain, inflammations, and is against clotting also. Okay, and scientists in 1897 from the Bayer company began studying the applications of acetyl salicylic acid in medications, and there is a flow chart which shows the different levels of discovery that has been taking place right from 1700 till date, because aspirin has several properties. It is used to prevent bowel cancer, then there is secondary prevention of stroke. It is found to inhibit platelet function, also found to inhibit cyclooxygenase, which is involved in anti-inflammatory response, and all of them. Okay, so it is known as weeping willow because the branches of the willow salic species are drooping down. Okay, so that's why the name is, and this is the medical formulation of aspirin and the bark that is used. Okay, the next drug of choice is digitoxin. Now, digitoxin and digoxin both are glycosides, steroidal glycosides. It's a cardiotonic drug, and 6 grams of digitoxin is isolated from a 10 kg of leaf sample through a series of process. And the chemical structure is also given. Now, both of them differ in having an addition hydroxyl group at C16 of the steroid skeleton. Okay, so species of digitalis such as digitalis orientalis, digitalis lanata, digitalis purpurea are sources of this drug. So this belongs to the foxglove family or Scrophulariaceae, which are producing a lot of cardioactive steroids. And the most prominent of this is digitons. And yeah, it differs from other animal-based steroids and all of the thing. So next one is morphine, which is produced from the poppy plant, that is Papaver somniferum. Both paclitaxel, that is Taxol, and morphine are drugs of importance that is used in hospitals and clinics for disease treatment. And both sources are from cultivated sources itself. So you have a beautiful picture of poppy, but it's also a narcotic drug, which is used to reduce anxiety, pulmonary edema, etc. This is a capsule from which the drug is extracted. Okay. Another plant of importance that has heralded a new drug discovery program is Taxol. Taxol was originally isolated as an anti-cancer drug from the bark of the gymnosperm, that is Taxus brevifolia. Okay, and the Food and Drug Administration of US as well as the European Medical Agency have approved Taxol for the treatment of several types of cancers such as breast cancer, ovarian, lung cancer, as well as Kaposis sarcoma. Okay, the metabolite produced is an belongs to the isoprenoid family, which is a diterpene, okay, which is formed by the condensation of 4 isoprene molecules. So this is the structure, and this is the concentrate of Taxol that is given for the treatment. And this is it's known as the Pacific U. So like that, there are many types of use. The European use there, Himalayan use there. Okay. And the biosynthesis of Taxol is a complex process involving 19 distinct enzymatic steps. And so you have taxane as well as Taxol as a product. Okay. Then I don't know whether this is very familiar to you. We have another drug called galantamine, which is actually a plant growing. Galantine is the plant. Galantamine is the drug produced by the plant Galanthus voronoi. It belongs to the Amaryllidaceae family, which is very close to the lily family. And this is otherwise known as the snowdrop plant because it is found growing in the cold regions of Russia, Turkey, and Iran. And there are other related species from which the drug is obtained. And because it is used in the treatment of Alzheimer's disease, this drug has so much of importance. See, when you pick up a slide, when you download the slide from the internet, you should be very careful because many times it is misnamed. Okay. Somebody has posted something like daffodil. Daffodil's name is Narcissus pseudonarcissus. And it was wrongly labeled as Galanthus voronoi. Okay, so we should be, I want to just send you the message that when you download things, you should be very careful while looking into the message. I hope you're all teachers. Are you all? Okay. Okay, so that's why galantamine crosses the blood-brain barrier. It is also used in the treatment of anesthesia as well as neuropathic pain in humans for ages. And you also have the significant plant-derived pharmaceutical products, that is aspirin that I've already told you, which has analgesic and antipyretic properties. You have atropine from Atropa belladonna, which is anticholinergic, then pupil dilating drug. Then digitalis, of course, we know diasporia, there is an anti-fertility drug that is diasgenin, which is the active molecule. Then we have anti-tumor and anti-gout agent, colchicine, which is obtained from Colchicum autumnale. So vincristine and vinblastine, the anti-tumor drug obtained from the Madagascar periwinkle called Vincarosia or Catharanthus roseus. So like that, there are so many products in pipeline, okay? Podophyllitoxin, camptothecin, artemisinin, which is also used as an alternate source of anti-malarial drug. Trichosanthins from one of the process of Trichosanthus, which we all use for as a source of vegetable. It has got anti-cancer as well as antiviral properties. Then Silybum marianum, the milk thistle. It is also used in treating hepatic disorders and is also used to treat the mushroom poisoning belonging to the group Amanita phyloides. Okay, and the list is endless. And now we know that among the plant sources, one second, I'm just scrolling this slide. Yeah. From the plant sources, there are several groups belonging to plant. You have the bryophytes, pteridophytes, gymnosperms, the algae, okay? So I want to concentrate on algae, which are also known as macro seaweeds, okay? And people living in the coast, they are well aware of the health benefits of the seaweeds, okay? Kerala coast, Tamil Nadu coast, Andhra probably, and Western world uses lot of salads in the form of seaweeds. The brown seaweeds belonging to Phaeophyceae, red seaweeds belonging to Rhodophyta, and green seaweeds belonging to Chlorophyceae are excellent source of health protection. They have anti-cancer properties, anti-inflammatory, immunomodulatory, neuroprotective, hepatoprotective. It works against cholesterol, so antiviral property, antioxidant, etc. So here I would like to project not all the properties, but one of the major aspects of the cell wall polysaccharides that are preferred in these seaweeds. Why they are preferred? Seaweeds do contain a wide range of cell wall polysaccharides in the form of dietary fibers such as albuins, the fructans, okay, and algin, so many of them, furcellular, etc. So it provides a flexible structure that helps us, okay, to digest the food or any other related problems. There are seaweed polysaccharides, mainly sugars and acids of sugar, which contain the sulfate groups attached to them. So they are referred to as sulfated polysaccharides, which have important binding properties. So taxonomically, they are very, very important. There are ficoidans, then carrageenans, algins, etc. And they are known to be proved sources of antioxidants, anti-tumor agents, anticoagulants, antiviral and anti-inflammatory agents. So all these are taken from related articles which have been published. And one application of these seaweeds is their antiprotozoal potentials. So there are many protozoal mediated diseases such as those caused by sandflies, then tsetse fly, and the protozoan parasite that is Leishmania species. Okay, and each one exhibits different kinds of symptoms. And over the past so many years, 28,000 compounds of marine origin have been isolated with hundreds of chemical entities and macroalgae, which range from 30,000. There are several species which have been documented from the coast worldwide, of which 151 have been investigated against the Leishmania parasites. And extracts, some of the study also reports that 48 species of macroalgae belonging to the brown algal family, that is Phaeophyceae, have been screened against Leishmania parasites. And little lesser, even Rhodophyta members, 80 species of them have been employed for studying the relevance of the extracts against the both kinds of Leishmania parasites. And we have one alga, brown alga, belonging to Dictyotaceae family. They are flattened seaweeds found growing in the rocky coast environment in the mid-intertidal zone. It is called Bifurcaria bifurcata. It is active against Leishmania donovani, the trypanosomal parasite, with IC50 values of 18.8 microgram per ml. Okay. So similarly, there is a triterpene derivative called fucosterol, which is being isolated from another brown alga, Lawsonia verdosa. Okay. And this has been proved to have a good IC50 value of 7.89 micromolar and 10.3 micromolar respectively at different vector stages of the protozoan parasites. So similarly, from England and Ireland coast, 21 brown algae have been employed to study their effect on the trypanocidal, leishmanicidal, and antimycobacterial activities also against Mycobacterium tuberculosis. And 4 extracts, it seems, were very prominent against Trypanosoma cruzi, etc. And again, there are 2 of them, Halotris silicosa and Bifurcaria bifurcata, which belongs to Dictyotaceae and Sargassinaceae, to be most potent with IC50 values of 1.2 and 1.9 microgram per ml. So this could be maybe in the next clinical trials, we don't know. So this is how Bifurcaria bifurcata looks like. The brown tuning fork seaweed with potential source of drugs. And this is the structure of bifurcatriol, triterpene terpenoid. Okay, so that's why now we have plants as one group of organisms which can have therapeutic effects with the advent of several new drugs that have been employed for therapy, whether it is anti-malarial, anti-cancer, etc. And health practitioners who practice Ayurvedic medicinal system, as well as the traditional Chinese medicinal system, do employ many of the plants with ethnopharmacological background to study several diseases. And therefore, plants alone cannot offer the inexhaustible supply of drugs for humankind. Okay, there should be some other process through which it can cut down, and then something has to replace the plants for the extraction of compounds, etc. So we need another strategy which needs immediate attention. So what are the alternative strategies apart from plants that can supply the inexhaustible resources of drugs? One is microorganisms. And we know through the years, so the discovery of penicillin, streptomycin like antibiotics. So penicillin is an antibacterial agent that was identified by Alexander Fleming way back in 1920s. And it's a broad spectrum antibiotic, the structure of which is the same. And therefore, based on this discovery, a group of scientists led by Gary Strobel from the University of Montana, Bozeman, Montana, US, applied isolating endophytic microbes. Okay, and it is like, he's like a goldsmith. Anything he touched has gone into the application level. I don't know how many of you are familiar with microbial endophytes. Definitely, Gary Strobel was so motivational. He derived the motivation from natural product chemists, Manush Gawani and Munro Wall. Okay, and they traveled to all places in the continent, whether it is South America, whether it is Australia, to unique regions to identify plants of ethnobotanical importance, and then isolate the endophytic fungi and the endophytic actinobacteria living inside the plant fragments. Okay, and that success story is today the first anti-cancerous drug from the microbial source, that is the endophytic fungi, Taxomyces chandrianae. That's a new novel fungus that was isolated in 1994 by Sturley et al., again Gary Strobel's student, along with the husband Donald Sturley. And therefore, this opened a new vista into bioprospecting of endophytes for pharmaceutical derived compounds. Okay, there are a number of endophytes that have been identified now, which produce the metabolites very, very similar to that of the host plant. The first story is about taxol. Okay, so I just want to put a slide for the endophytes if you are not very familiar. They are the microbes living inside the plant fragments, and it seems the medicinal property of the plant is because of these microbes. That's what is been contemplated now. There are methods of isolation. You give a surface sterilization with alcohol treatment, and then you plate the surface, sterilize them completely, wash in distilled water, and then plate them in the minimal media. Water agar is the medium that is used for plating of fungi from the plant fragments. And we have actinomycete isolation medium for the isolation of endophytic actinomycetes. So you obtain the cultures, then catalog them. Then you have microbial fermentations, where you can use elicitors or produce a compound or take them for higher level of application. So that is the endophyte-based techniques, okay? And we did an invited review from Springer, wherein we actually gave the entire 52-page or 60-page write-up about novel metabolites of endophytes, both actinobacteria and fungi. And we have seen that endophytes could be divided into the fungal endophytes and actinomycetes endophytes. Both of them have unique properties, biological properties or bioactivities, and some common bioactivities. So this is taken from my publications. So now I'll be discussing about the host-related novel bioactive metabolites produced by the fungal endophytes of medicinal plants, which include taxol or taxane production, camptothecin, podophyllitoxin, hypericin, vincristine, and vinblastine. There are others. I've taken only 5 or 6. Okay, so paclitaxel or taxol, as it is named in the trade, one of the most exciting discoveries in science, because from the bark of the Pacific yew tree, that is Taxus brevifolia, natural product chemist Guani et al. Identified the drug. So it seems to treat a patient, you need 2 grams of the drug, okay, which is otherwise you have to cut down almost 12 large yew trees, okay. And this unique drug, which is of anti-cancer importance, has the ability of bringing down the depolymerization of tubulin during cell division, and therefore it is very, very effective and all for breast cancer cell line, ovarian cancer cell line, lung cancer sometimes, etc. Okay. Now the milestone was achieved when one of Gary Strobel's student, Andrea Sterling, identified this unique fungus Taxomyces chandrianae from the bark of the Pacific yew by isolating and plating them. But the drawback of this fungus was the low production of taxol, and therefore the focus was shifted from Taxomyces chandrianae to another fungus of the rainforest. It's known as Pestalozziopsis species. Pestalozziopsis microspora was isolated from Himalayan yew, Taxus wallichiana, from one of the students who was from Nepal, and it can produce the taxol. That is, this fungus, Pestalozziopsis microspora, has the ability to produce 60 to 70 microgram of taxol in per liter of the medium. Okay, and from several sources, it's been identified. So this is how the taxol plant, the fruiting body looks like. This is the fungus Taxomyces chandrianae, and this is the structure of the paclitaxel. So now we have another producer, that is Pestalozziopsis microspora. This picture is taken from my publication in Springer book, and first anti-cancer compound. Now the colonies of the fungus, it is pinhead cushion-like. The acerulus produces black oily ooze. So if you take that and then mount them, you have millions of fish-like spores, conidogenous cells with branched appendages. That is the identification of the Pestalozziopsis species. Okay, and this is the electron micrograph. It's called as the rainforest endophyte because it's been isolated from almost all endophytic, from all the rainforest plants around the world. There are several, a high level of generic and the structural diversity is represented, or species diversity is represented by this Pestalozziopsis. Okay. Next is camptothecin. Camptothecin, also abbreviated as CPT, is an anti-tumor chemotherapeutic agent obtained from the bark of the medicinal tree, which is of Chinese origin, called Camptotheca acuminata. And again, by the natural product chemist Walls group, they have identification of anti-tumor agents 1, 2, 3, 4, 5, 6, like that they have published. So it has got the efficacy to treat various cancers, but since the drug has lots of side effects, this was discouraged from the clinical studies in 1972. And because of its DNA isotopomerase activity and as a cellular target, it's again back into action by 1980. So there is a potent alkaloid that is produced by the endophytic fungus isolated from Notipodidis foetida, another name for Camptotheca accumulata by Puri et al. In 1975. The producer fungus is Enterophora infrequens. Okay, and cytotoxicity assays have given good results. Similarly, there is another fungi which is called Fusarium solani, which can also produce analogs of camptothecin, that is 9-methoxy and 10-hydroxy camptothecin, okay, which was collected from the Yunnan province of China, one of the biodiversity hotspot location. Okay, so this is also the both 9-methoxy camptothecin and hydroxy camptothecin are precursors for the synthesis of synthetic anti-cancer drugs, topotecan and irinotecan. Okay. Similarly, we have another example of hypericin. Hypericin is an antidepressant agent because of its monoamidase activity, and this is obtained from Hypericum perforatum, otherwise known as St. John's wort. Okay, so we have Kusri's group, like Strobel, Kusri, and Spitaler from Germany, have successively identified most of the bioactive endophytes from all over the world, and they have a good collaboration. Many books have been written, and it's been used at even university levels as research grants to obtain projects on endophytes. So there is a fungal endophyte called Chaetomium species, Chaetomium globosum, which has been collected from the Hypericum perforatum plant growing in wild along the Jammu and Kashmir regions of India. So this fungus has the ability to produce hypericin as its as well as its precursor amodin at so much of quantity under fermentation conditions. Okay. Next story is about podophyllatoxin. It is also one of the important anti-cancer agent because it has the ability to suppress the formation of mitotic spindles and microtubules during the cell division. And therefore, two species of podophyllum, the American species as well as the Indian species have been studied to produce this drug. We also have two fungi which have been isolated from Podophyllum peltatum as producers of this podophyllatoxin and their related lignans. The last, but not the least, we have vincristin and vinblastin produced by the Madagascar periwinkle plant known as Catharanthus rosea or It's otherwise there's 2 or 3 names associated with this, Lochnera rosea, etc. So vincristine and vinblastine are alkaloids which were discovered in 1950s by this pharmaceutical company, Eli Lilly. Okay, and another research group from USA and Toronto. And it's been immensely used in cancer therapy as a source of alkaloids for treating cells, cancerous cells. We also have fungus, Fusarium oxysporum, which produce the same kind of alkaloids, vincristine and vinblastine, at so much of the quantification in the culture media. Okay, the story is endless. But now I would like to project another group of microorganisms called as actinobacteria. I don't know how many of you are microbiologists here. Are there any microbiologists?
Yes, madam.
Okay, fine. What do you, oh, Niranjan Raj. Dr. Niranjan Raj. Okay, I know you. I hope my presentation is useful to you.
Very much, madam.
Okay, so actinobacteria are a group of endophytic microbes or soil microbes which found its importance in discovery of metabolites by the production of streptomycin by one of the strains, Streptomyces griseus, identified by Waxman. Now, Waxman is like strawberry. Whatever he touches, it's like gold. So he has identified several antibacterial compounds from unique actinomycetes. There is also a genus called Walkersmania, which is actually given in his honor because he has obtained so many or given so many important contributions. So why is actinobacteria? It's a phylum now. So in the classification, it is elevated from the class to phyla of actinobacteria and includes 6 classes, 18 orders, 14 suborders, 63 families, and 374 genera as per this database. Okay, and they are prolific producers of antibiotics. And we know of the approximate 70,000 microbially derived compounds, 29% is contributed from the small group that is actinobacteria or actinomycetes, they are called. And approximately 60% of antibiotics produced from actinomycetes between 1950s and 70s were exclusively from the genus Streptomyces. Okay, so that's its importance. And I have highlighted the discovery of the streptomycin antibiotic by a group of co-workers, that Waxman leads the group, and in 1952 who got the Nobel Prize for Physiology and Medicine for his discovery of the antibiotic streptomycin. So generally actinobacteria are distinguished from fungi by their powdery colonies, and they have very slow growth. Okay, so this is the structure of broad spectrum antibiotic, that is streptomycin. So based on these inputs, now we go into marine rare actinomycetes, a promising source of structurally unique and diverse novel compounds. Okay. Now the term rare actinomycete is given to those actinomycetes other than streptomycetes. So you can include Pseudonocardia, Micromonospora, Nocardiopsis, Deetsia. There are so many of them, and there is so much of identification problem in actinomycetes. You have to go for the DNA isolation and sequencing of the 16S rRNA gene, and then deposit them in GenBank, blast search, and then identify. That is where your bioinformatics comes into picture. Okay. And so far, compounds produced by marine rare actinomycetes represent several activities such as antibacterial, antifungal, anti-cancer potential, anti-malarial, anti-parasitic, etc. Highest number of natural products obtained from the following genera, that is Micromonospora, Salinospora, Pseudonocardia. I don't know how many of you are familiar with this name, Salinospora. Now, this is a novel actinobacteria that has been isolated with over 2,500 strains from the marine environment. Marine environment means from the ocean at a depth of 1,100 meters below. There is a group, William Fennikel's group from USA. They have traveled, they have sampled, and they have isolated 2,500 strains, of which there is a metabolite, cellulose-poramide, which is a definitely anti-cancer and cytotoxic agent. There are 7 variants in there. That is one. So now also this has been developed into drug. Okay, it's an approved drug. Or one of they do it's in it is in the phase 3 chemical trials. Okay, clinical trials. So that's how these organisms are. And we have several reviews that have been given. So one of that is novel natural products from marine Micromonospora species. So there is a label here. It produces a macrolide that is anti-proliferative. Then no problem, we have another polyene macrocyclic lactam, but there is no activity for that. There is a cytotoxic metabolite, many of them antibacterial diterpenoids, aromatic tautomer with no activity. Then we have antibacterial agent, butyrmycin. So like this, the list goes on, okay? And unusual biologically active compounds are produced by marine rare actinobacteria. So unusual compounds, that means they are novel, not reported so far, okay? With high structural diversity. So we have Actinomadura species, Actinoallotechnus, Nocardiopsis species producing so many activities. One is acetyltransferase inhibitor, like a triphosphate inhibitor, then anti-candida-like activity, anti-parasitic, anti-malarial, anti-allergic. So these are the potentials of actinomycetes. Now, there is a saying always, new species of actinomycetes do not always produce new compounds with high frequency. This has been published in the Journal of Antibiotics by Japanese workers, Takagi and Shinya in 2011. Now, we know that the actinomycetes are immense source of bioactive compounds, but rate of discovery has been decreased in the upcoming years. So what these authors have done is they have isolated the actinomycetes from several areas, endophytes, plants of medicinal plants of origin, then from the marine environment, from the soil, etc. So of the 106 new species that have developed, 7 strains belonging to Streptomyces produced 10 new compounds. Okay, and there is a list to anthracycline. Everything is new. That's why in my presentation, to understand, see every line, if you just type and then keep it in the white color, you will not understand the importance of presentation. Probably if you color your or differentiate your talk in different colors, you'll understand the importance. Okay, so wherever there is new, I have given different sorts of colors to distinguish. And there is a table here which says the producers of new compounds. So from their study, they have isolated 10 new species. Not necessarily, they have it from marine sponges, mangrove soil, lichens, bark samples, etc. And these are the unique compounds that have been produced from these actinomycetes. So known species isolated from soil, from the mangrove environment, that is marine environment, marine sponges, etc., have produced also new compounds. So not necessarily new ones should produce the compounds, even the already known ones also can produce newer compounds. That is due to the chemical diversity. So why has marine natural products gained so much of importance? So let's see this write-up. So marine natural products have been discovered, and they are still being discovered. And it is because of the research that was done way back in 1955, quite a few years back by Perchman and Burke, wherein 2 new cytosine nucleosides have been isolated from the sponge species, spongogoridin and spongothymidine. Okay, so till then, it was not known that nucleosides with sugar residues were found to be bioactive till then. So that paved the way for isolating newer compounds from marine environment, including the sponges. And that's why the first drug from the marine biology side is cytarabine. Okay, it's a cytotoxic agent that's been isolated from the sponge Cryptotheca crypta. Okay, this is from a Caribbean sponge. So that completely changed. Then we have several other novel compounds that have been isolated from marine sponges, and more than 28,600 marine natural products have been obtained as a result of combinatorial chemistry, either by derivatization or new synthesis. Okay, and that's why there is a more and more quest for the isolation of compounds from marine natural products. So this table clearly shows that some of the agents are in the pipeline for the, there are some statistics which has been given. Marine environment, not necessarily you have to take the marine sediment, marine water, or there are other invertebrates which live in the marine environment, the sponges, the bryozoans. Bryostatin 1 is one such compound. So like that, there are several of them that have been identified in the past years. Tunicate, then a herbivorous mollusk called the seahead, Dolabella auricularia, from the Indian Ocean, is also the source for the production of 15 cytotoxic cyclic and linear peptides, the dolostatins, of which one, that is dolostatin 10, has been chemically synthesized and is currently in phase 1 clinical trial. So this is the potency. There are also sponges from other part of the world which have been sampled for bioactive metabolites, and one includes helicondrin B, a macrocyclic polyether, which is isolated from Helicondria ocadi, which is a novel molecule in 1985, and currently it is in the preclinical trials by the National Cancer Institute. So Helicondrin B has also been isolated from some other marine sponge and is also grown in New Zealand territorial waters. So like that, we have the discodermolite produced by Discoderma species, and eleutherorobin isolated from both. Erytherophobia are similar to that of paclitaxel. It's also an anti-cancer drug producing Caribbean sponge, and Novartis is a company which has taken up the clinical trials from it. Okay, so here's a list of sponge-derived compounds. We have spongothymidine, spongouridine, the first compounds produced, and then cytarabine, the marketed anti-tumor agent. So like that, we have several variations, vidarabine, then nelarabine, fluorabine. So all are nucleosides with sugars attached, and several marine-derived licensed drugs are under clinical trials. So these are candidates. It's been taken from a list modified by Pereira et al. In 2019. And there is a journal called Marine Drugs where such information reviews are available for your kind attention. And here we have a list of marine-derived chemotherapeutic pipeline, chemotherapeutic drugs. So we have 4 approved drugs, of which cytarabine from the sponge, Tetia, tops the list. So we have the targets and the cancer conditions which the drugs are treated. Then you have trabectin from a tunicate, then helicondrin B from the sponge, then dolostatin again from the seahead. Okay, so like that, there are drugs which are also in phase 3, phase 2 as well as the phase 1 trials. The names have not been decoded, but their molecular targets have been achieved. Okay. Now we also have drugs from marine invertebrates which are under clinical trials. This is from a different paper, but more or less same information is available. We have bryostatin, didymin, dolastatins, again produced by marine species. You can classify them. They are ascidians, bryozoans, cyanobacteria living in sponges, then molluscans, etc. So you cannot underestimate any organism. Each have the ability to produce the prescribed compound. Now, I would like to end my talk by concluding that natural products, they are a promising tool or a pool of diversity of chemical structures wherein the bioactivities can be directly associated with. Natural product-based discovery also offers a unique niche for diverse forms of academia-industry collaboration, and based on this, certain virtual consortia, such as the International Natural Product Sciences Task Force, has been implemented. It also provides a platform for integration of expertise, technology, and materials from the participating academic as well as industrial entities. And though we have several constraints in the production of natural product drug, because of its accessibility, then sustainability, IP constraints, and as well as sharing up of patents because of the biodiversity rights. Marine environment is not limited to one country. There are countries which share borders with the ocean. So that where the IP constraints take place. And these are the issues which have to be followed according to the CBD, Convention on Biological Diversity, as well as following the Nagoya Protocol, Kyoto Protocol, etc. So therefore, the scientific and technological advances provided here offer a strong basis for drug discovery to make a major contribution to human health and longevity. That's the theme of our refresher course. And I would like to conclude my talk citing this reference, which was given by Lai and Vaderas in 2009. It was published in Science. Drug discovery and natural products. Is it an end of an era or an endless frontier? There is no full stop for this. As long as microbiologists, natural product chemists, and biologists are there, the search goes on. Okay, thank you. So if you have any questions, if it is in my purview, I would like to answer.
Ma'am, good afternoon, ma'am.
Yeah, good afternoon.
Ma'am, these isolated constituents only determine the medicinal properties of the plant or seaweed or any other natural source, ma'am. Since time immemorial, we have been using this plant material to cure various diseases.
Yes.
While the time we are using the crude extract only. But that time they cured so many diseases. But these constituents only determine the medicinal properties of the plant or
it is the constituent that determines the medicinal property of a plant. If you go through ethnopharmacology journals, the system is there, okay? It's a cure, but you need an active ingredient. Unless the active ingredient is there, whatever you take, it cannot be 100%. So that's why you have a drug formulation involving a synthetic molecule as an active ingredient. And then the rest of it is potato.
Yes, thank
yeah,
you.
for everything. It's not that. Even we use turmeric, we use so many spices. Indian spices are rich in medicinal constituents. Yes. Are we free of diseases? I don't think so. There are so many constituents present in the crude extract. If you have an individual constituent, it acts particular accordingly. Okay. For example, curcumin is present. Is it in the bioavailable form? So what happens? Is it, see, if you make it dissolve in water or alcohol, is it more soluble in alcohol? So like that, that is one example I'm giving you. We use curcumin in our food in the form of turmeric, okay, or apply to the face or anything. It may give us a topical application for eradicating or enhancing your complexion, etc. But people also have so many diseases associated even after using the medication, right? That is what. Every drug should be in its capsule form or tablet form where the active ingredient is there.
Thank you, ma'am.
Yeah. Niranjan Raj, Dr. Niranjan Raj has no questions.
Madam, I have one question, madam.
Yeah, tell me.
Can you please, let us know the status of this genetic modification of plants in terms of this drug molecule identification?
No, I am not aware of this. Genetic modification of plants itself is a different topic. For the drugs, okay? You can't modify a plant for a drug. You can modify a microbe. You can include a biosynthetic gene cluster into the microbe and make it produce the metabolite of importance.
Okay.
So what you're talking is of GM crops, those are different, okay? So if a plant has a gene for the biosynthetic cluster for producing the metabolite, probably you can introduce that to another plant like your, the yeast expression system or heterologous expression system, and then make it produce the enhancement. Otherwise, you cannot, or through cell culture means. So like that, production of a particular metabolite, that is possible. Yeah.
So, good afternoon, ma'am. Like you were speaking a lot about plant products and its medicinal uses, and I would like to just ask. Why research has been banned for this particular, about hemp, cannabis indica or cannabis sativa?
Yeah.
But there is a great lot of potential, but in India it is allowed, but in few countries like in USA it is used as a medicine purposes, and also there are few products that have been developed. But why not research? Just basically because they are drugs, exhaustive drugs, like for other purposes, so they are not allowed, or is there any particular thing that is being making?
We all know that these belong to the, especially opium, then there are 2, 3, heroin, and all of them are narcotic drugs. There is a way that you give it as anesthetic or pain reliever, etc. But what happens when you take, it takes control over your brain, okay? And therefore, you want more and more. So then it becomes abusive, right? A drug becomes so abusive when you are addicted to it. That is what is happening in the case of the opium, your cannabis, and all of them.
Do you believe that most of the drugs are becoming addictive? Like even sugar is also said to be an addictive one. One of the
one thing I will tell you. I was diagnosed under stress for diabetic condition. I had a value of 250 after food. Okay, how do you bring down the sugar level? Tell me. Either you must go for diet.
Glycomet, diet or glycomet.
Otherwise, there are different kinds of medication. You could go to take insulin, or you have the oral alpha malase, alpha glucosidase inhibitors. You correctly give the doctor the advice and ask him to not to put you on insulin. And then I got it down to 97 now by following strict diet. So what do you say? Only with diet, it cannot be. Only with tablets, yes, it can be. Okay. You just take the insulin plant or something else and then eat, and then you say, I'm going to get it. I don't think so. So medication is important.
So you don't believe in reversal of any of these conditions, right?
No. I am now drug free. You understand?
Yeah. Okay.
only because
Okay.
of my achievement, my control in diet, and then
Yeah.
following the doctor's advice properly. This is the advice that we are going to give to everybody. No
That's
disease
true.
is for life, okay? It's in you to take over the disease and then be disease free. That's
That's
what.
great. That's a great advice. Thank you, ma'am. Thank you very much for the
Yeah, I
informative
hope
session.
the presentation was useful to you all. Okay.
Yes, ma'am.
Yeah, ma'am,
Yes, ma'am.
absolutely.
Yes, ma'am. It is very useful. Ma'am,
Thank
it was
you.
very highly informative.
Huh?
It was highly informative, ma'am.
Okay, thank you. With the little time I had along with classes, I was able to make these slides. It will be shared through the coordinator. Okay? Yeah, good luck to you all.
Yes.
Like
Thank
you
you,
were
ma'am.
also
Thank you.
participants a few years back, and now I'm a resource person here. That's what. Okay, thank you.
Thank you, ma'am.
Thank you, ma'am.
Thank
Good luck. Good luck. Please be online.
Yeah, participants, please join at 1:50. Thank you.
Okay, ma'am. Thank you.